Hi, I'm Oshri Avraham from the laboratory of Dr. Avihu Klar at the Medical Neurobiology Department in the Hebrew University of Jerusalem.
Hi, I'm Sophie Zisman and I'm also from Avihu Klar's lab.
Today we're going to show you a procedure for Ovo Electroporation and spinal cord open book preparation.
We use this procedure in our laboratory to study axonal trajectories of genetically defined neuronal population in the embryonic spinal cord.
So let's get started.
To begin this procedure, transfer fertilized white leghorn chicken eggs from room temperature to a humidified incubator, preferably with rocking trays at 37 to 38 degrees Celsius. It is important to monitor the temperature and humidity of the incubator in order to obtain viable and synchronized embryos. The chick embryos are electroporated when they have reached Hamburger and Hamilton or HH stage 18 to 20, which is an optimal stage for enhancer-dependent specific expression. At this stage, the head lies at right angles to the trunk. Also at HH stage 18 to 20, a vast set of extraembryonic blood vessels, such as anterior, posterior, right and left vitelline vessels, should also be seen. The chick embryos reach HH stage 19 to 20 after 66 hours of incubation, and at that time remove the eggs from the incubator. After removing the eggs, place them horizontally and wait for 5 to 10 minutes. The embryo is now located at the upper pole of the egg. Prior to electroporating the embryos, prepare Hank's solution with 100 units per milliliter of penicillin and 0.1 milligrams per milliliter.